Good morning and welcome to the AltruBio R&D Day. At this time, all attendees are in a listen-only mode. A question and answer session will follow each formal presentation. If you'd like to submit a question, you may do so by using the Q&A text box at the bottom of the webcast player or by emailing your questions to questions@lifesciadvisors.com. As a reminder, this call is being recorded and a replay will be made available on the AltruBio website following the conclusion of the event. Before we begin, I would like to remind everyone that today's conference call will include certain forward-looking statements. These forward-looking statements are not guarantees of future performance, are subject to various risks, uncertainties, and assumptions, known or unknown, which could cause actual results to differ materially from those indicated or anticipated, and speak only as of the date of this call. I would now like to turn the call over to your host, Judy Chow, President and CEO of AltruBio. Please go ahead, Judy.
Hello, everyone. This is Judy Chow. Please progress to the next slide. It's a pleasure to introduce you AltruBio. AltruBio is a company focused on the novel therapeutics for immunological diseases. Next slide, please. Today, joining me are Dr. Jesse Ho, our Chief Medical Officer at AltruBio. Also, Dr. Samin Abedin, who is the Assistant Professor at Medical College of Wisconsin. Next slide, please. Today, we will go over the company overview and the science background, and we'll share with you our lead molecule, ALTB-168, the clinical proof of concept data, and Dr. Abedin will dive deeper into the treatment refractory AGVHE data, which we just published at the ASH conference. Then we'll introduce you our second generation molecule, ALTB-268, and to share with you the pipeline in a product potential. I will wrap up the presentation by sharing with you our path forward, and we'll be happy to take any question you have. Next slide, please. Let me first begin with the company overview and our science. Next slide. The reason why we're so enthusiastic at AltruBio because we have the first-in-class mechanism of action. Our research team is the first research group identify PSGL1 plays a key role in regulating late-stage chronically activated T cells. And with this knowledge, we have developed our lead molecule, ALTB-168. 168 so far has been validated clinically to close to up to 200 patient data in 4 different indications. And with this profound proof of concept, we have to build further, expand the potential with our second generation molecule, 268. 268 is going to be developed into a subcutaneous dosage form, and we are about to enter the clinical phase to further validate the molecule. 268 has a higher potency than 168, so it definitely can expand all the potential 168 has already proven. And we are encounter a lot of significant milestones, and including recently the IND acceptance of 268 and the publication of 168 at the ASH conference. And we're moving forward also for 268 to enter the phase 1 and also phase 2 at the second half of 2023. And this science has been managing under the very experienced management team in the past couple of years. And with strong track record clinically and commercially, we like to bring this new treatment and mechanism action to the patients as soon as possible. Next slide, please. Our 2 lead molecule, 168 and 268, has been demonstrated in downregulating late-stage chronically activated T cells. Both molecules serve as agonist antibodies to enhance PSGL1 function. PSGL1 has been reported and demonstrated among different kind of research team, showing it's a key immune checkpoint regulator. So 168, 268 is further enhance this immune checkpoint function when PSGL1 expression is higher in the activated T cell stage. And then 168, 268 can enhance the function to bring down the regulation of T cells during the immunological disease stage to bring the overactivated T cell down, bring back the immune balance and the immune so-called homeostasis. Important to point out, our research team has been focusing on 168-268 regulating PSGO1 function without any interference to the PSGO1 role in cell migration. With that background, next slide, please. I'd like to turn over to Jesse for him to walk you through the proof of concept we already obtained from multiple different indications. Jesse?
Thank you, Judy. So my name is Jesse Hall. I'm the Chief Medical Officer of AltruBio. I joined Judy and the management team here approximately a year ago, and I'm very excited to present some of the progress that we've made over the last year. Next slide, please. Although 168 has a novel mechanism of action, it's clinically validated across a broad range of autoimmune disease and has demonstrated safety and tolerability across this wide patient population. Following the initial first-in-human studies, an initial study was done in the psoriasis patient population. And in that patient population, dose-dependent decreases in disease activity were observed. Continuing on from the success from that psoriasis program, the molecule was then moved into psoriatic arthritis. And it was dosed up to 9 milligrams per kilogram for up to 12 weeks in a patient population with moderate to severe psoriatic arthritis. And I'll be sharing some of that data in a subsequent slide. With the completion of the psoriatic arthritis data, a study was initiated in a patient population with ulcerative colitis. The drug was dosed at 9 milligrams per kilogram for up to 12 weeks, and the standard outcome measures were measured. And if you look at the data coming from that study, had this product been more patient-friendly in its approach, meaning a subcutaneous injection, likely the company would have progressed this into later stage studies. But given the fact that the therapy was administered as a weekly IV administration, the company believed that the best next step would be to move it into a patient population where IV administration was more amenable. And this is some of the data that Dr. Abedin will be presenting from our recently completed acute GVHD study, in which patients were dosed with 6 milligrams per kilogram as a starting dose, followed by 3 additional doses of 4 milligrams per kilogram. We are very excited to present that data at the ASH conference and happy that Dr. Abedin will be presenting that today. In addition, Dr. Abedin is leading a frontline study in acute GVHD with 168, and to date we have seen some promising results. Overall, over 200 patients have been dosed with 168, and we're quite confident that this provides a good option for patients with acute GVHD. Moving forward, our plans for 168 is to identify a potential partner while we focus our internal resources on the development of 268. Next slide, please. This is some of the data taken from the psoriatic arthritis study. This was an open label study, a phase 2 study in patients with active psoriatic arthritis. 20 patients were dosed with 168. Effectively, weekly dosing was administered initially, followed by bi-weekly dosing. The primary endpoint was at week 12. And at week 12, the ACR 20, 50, and 70 evaluations were conducted. If you look at the data on the left-hand slide, you can see that this data is competitive with current biologic therapies. And specifically, if you focus on the patients on the right-hand side, the 15 patients that received all 7 doses, the results were quite competitive with current biologic therapies. The challenge, again, with this therapy and this indication, it was an IV administration. And we believe the IV administration is better suited for GVHD while we focus on progressing 268, a potential subcutaneous administration into the clinic. Next slide, please. This is data taken from the phase 2 study in a patient population with ulcerative colitis, moderate to severe ulcerative colitis, that were refractory to biologic therapies. In my experience in conducting ulcerative colitis studies, this is the most refractory patient population that I have seen, with over 60% of patients being refractory to 3 or more biologics. In this study, 14 patients were dosed for 9 milligrams per kilogram for up to 12 weeks. The standard outcome measures were assessed at the conclusion of 12 weeks, including clinical remission, endoscopic improvement, histologic remission, and clinical response. We're comparing the 168 data in the teal green to data taken from the varsity study. So the varsity study was one of the first and only biologic studies looking at an active comparison between two biologic agents. In that study, adalimumab or Humira was compared to vedolizumab or Entyvio. The assessment was done at week 14, so this is a similar time point. And you can see across the broad range of outcome measures that 168 compares quite well to both adalimumab as well as vedolizumab. This is why we're so motivated to move 268, our next generation molecule, into the clinic. We believe we have competitive data in a biologic field, especially in a patient population with biologic refractory disease. And we are going to be initiating studies with 268 in the coming years. With that, what I'd like to do is turn the call over to Dr. Abedin. Dr. Abedin is an assistant professor at the Medical College of Wisconsin. He's quite familiar with 168 in not only the acute GVHD setting, as well as the frontline setting. We recently presented some data at the ASH meeting, and Dr. Abedin will be presenting that data today. Dr. Abedin?
Yeah, hi, good morning. My name is Samim Abedin. I'm a leukemia and bone marrow transplant physician at the Medical College of Wisconsin. And it's my pleasure to speak today about treatment refractory acute graft versus host disease, why this represents a significant unmet need for our patients after allogeneic transplant. Next slide. So I just wanted to begin by going over how allogeneic transplant, first of all, represents a potentially curative treatment for a number of high-risk hematologic malignancies. And through a couple advancements, first with the use of reduced intensity conditioning, second with increasing access to alternative graft sources, the number of allogeneic transplants performed in the United States continues to climb. So around 2020, about 8,800 allogeneic transplants were performed. This number, while it looks like there's a slight plateau on this curve, is continuing to increase with anticipated 10,000 allogeneic transplants underway in the United States in the next 5 years. Next slide. During an allogeneic transplant, patients receive stem cells from an alternative source, not from themselves, and that would be an autologous transplant. During the transplant procedure, these stem cells grow a new immune system in the body. And so graft versus host disease actually represents the most frequent complication after allogeneic transplant and represents the new immune system attacking its host. There are two forms of acute graft versus host. There are two forms of graft versus host disease. There's acute graft versus host disease. This is typically seen within the first 100 days of transplant, but with reduced intensity transplant can be seen up to the first 6 months after transplant, and occurs anywhere between 30 and 50% of patients. Chronic graft versus host disease typically performs later after allogeneic transplant, and up to 60% of patients typically manifest between 6 months and 2 years after allogeneic transplant. Next slide. So in terms of acute or chronic graft versus host disease, acute graft versus host disease represents the type of graft versus host disease with the greatest amount of morbidity and mortality associated with it. So a significant concern for allotransplanters. It's thought to occur due to host tissue injury, and this is due to the fact that when we do an allogeneic transplant, we administer either chemotherapy or radiation to eradicate any sort of residual disease. This ultimately results in some degree of tissue injury. And then subsequently, upon engraftment, these T cells see this inflamed environment, resulting in T cell activation, ramp up of signals of inflammation, and then subsequently graft versus host disease. And so Recently, you may have heard that there have been some measures or some treatments to improve prophylaxis against acute graft versus host disease. That being said, due to the nature of how acute graft versus host disease develops, we still see, even with advancements in prophylaxis measures, a similar incidence of acute graft versus host disease due to the nature of unavoidable tissue injury and T cell activation during the peritransplant time. Next slide. So for acute graft versus host disease, for the most part, initial management is corticosteroids. There are clinical trials that add agents to corticosteroids for first-line treatment for acute graft versus host disease. Upon administration of corticosteroids, responses are seen typically in about 50% of patients. And so if patients respond, they taper steroids. During the taper of steroids, sometimes there are flares. And so these are either considered to be steroid dependent or steroid refractory cases. During administration of steroids, there's a subset of patients for which there is no response or there's progression, and these would be considered to be steroid refractory cases as well. Next slide. So for steroid refractory acute graft versus host disease, we have a number of agents that are available for its treatment, but with recent data related to ruxolitinib, ruxolitinib has been considered in many institutions as the first-line treatment for steroid refractory acute graft versus host disease. On this list, other agents that are occasionally considered for the treatment of steroid refractory graft versus host disease include ECP or extracorporeal photopheresis. mTOR inhibitors, sirolimus are sometimes in use as well. But predominantly, in the survey of multiple large academic centers, ruxolitinib represents now sort of the standard first-line agent for steroid refractory acute GVH used in 80% or greater institutions as far as the first-line option. Next slide. So ruxolitinib, just to give a little bit more information, was approved as treatment for steroid refractory graft versus host disease based on the REACH trials. The REACH trials randomized ruxolitinib administered at 10 milligrams twice daily against best available therapy, for which ECP and ATG were the most commonly used options. The primary endpoint was overall response at 28 days, and what was seen was an improvement. So with best available therapy, steroid refractory GVH responded about 30–35% of the time. With ruxolitinib, these responses were seen about 60% of the time by day 28. Next slide. But what you can see overall is, in terms of the durability of the response, about 40% were able to remain in a durable remission or response with ruxolitinib. Certainly, in time, there is an increasing population that subsequently reached a treatment failure state. Next slide. So in the supplemental appendix for ruxolitinib in the REACH2 study, what you see is certainly in time, there was the need for additional systemic acute GVHD therapy. This is the treatment refractory sort of indication. And in this study itself, this was about 25% of patients. In terms of patients who potentially met this category, these most often represented more severe acute graft versus host disease, so grade 3 or 4 disease, GVHD involving the liver, or GVHD involving the lower GI tract. So while ruxolitinib has certainly served as a breakthrough agent in the management of steroid refractory GVH, what we are now confronted with is this increasing need for next line therapy, or this treatment refractory space, which potentially represents about 30% of patients who develop chronic or steroid refractory acute graft versus host disease. Next slide. Forward. Yes, thank you. So what do we have available? So unfortunately, we don't have great options for patients who fail ruxolitinib. So really what we're left with are agents that we previously used in the setting of steroid refractory graft versus host disease. So in the absence of trial, and there haven't been a large number of trials that have enrolled this population, common options that are turned to include ECP, TNF-alpha antagonists, sirolimus, Next slide. But we have very limited efficacy in this setting with these standard agents. To speak to that, what I wanted to point out was this US multi-center real-world analysis that was led by our group, which included a number of large academic centers that do allogeneic transplant within the United States. We identified 48 patients who received ruxolitinib and were defined to be resistant. And so there were criteria that were set by a consensus group that was published in the journal Blood. And so we identified patients who were quote unquote ruxolitinib resistant. And we wished to see how they did, with our best available therapies. So again, this makes sense. These are patients who in the REACH2 trial didn't particularly do well or didn't have full responses with ruxolitinib. So lower GI GVH, liver plus minus GI or skin GVH. And what we saw was when next available therapy was used, the responses were seen in about 36% of patients, so very modest. In terms of looking at this table, certainly in some patients, ruxolitinib was continued in that population. There was a slightly better response rate. But overall, this is certainly an area of emerging need within our population. Without control in the majority of patients, we saw very poor survival when patients were deemed to be ruxolitinib resistant. And we observed a median survival of about 28 days in this population, with the primary cause of death due to graft versus host disease. And so in this population, certainly what this represents is a great unmet need for our patients. Next slide. So with that, I'll introduce the nahilizumab trial, which we recently completed in steroid refractory and treatment refractory acute graft versus host disease. So initially, this trial enrolled patients with the objective of defining pharmacokinetics and defining initial response to agent. And the way that was performed was we initially enrolled patients to a single dose of nahilizumab with pharmacokinetics performed. And we enrolled patients specifically with steroid refractory skin acute GVH, so we would be able to clinically determine whether there was a response in these patients. If we had seen both a favorable pharmacokinetic profile, safety profile, and responses within our skin patients, then this trial would subsequently move on to multi-week dosing, and also within patients without just skin steroid refractory graft versus host disease. And so the second phase of this trial enrolled patients at a weekly dosing, and it enrolled steroid refractory acute GVHD. And then subsequently, this trial was amended to also include treatment refractory, which was defined as steroids and one other treatment, acute graft versus host disease. Our primary endpoint, again, was pharmacokinetics, both with the single dose and with the multi-dose schema. And we also then looked our secondary endpoints, which included efficacy, which we'll discuss, PK profile, and receptor occupancy. So next slide. In terms of enrollment, we initially enrolled 7 patients with steroid refractory at the 3 milligram per kilogram dose, 6 patients at the 6 milligram per kilogram dose. Next slide. And then for the multiple phase dosing, we enrolled 12 patients with steroid refractory acute graft versus host disease. So again, these are patients who initially received corticosteroids and then had either persistent or progressive acute graft versus host disease. We enrolled 12 patients with treatment refractory acute graft versus host disease. And so again, this is patients who had received steroids and had steroid refractory disease, had received another agent for which ruxolitinib was the primary agent that was administered, and then had either progressive or persistent disease. And so we had an equal enrollment in both the steroid refractory and treatment refractory indication. Next slide. In terms of patients that were enrolled, really the patient characteristic to point out in both the steroid refractory and treatment refractory cohorts, we enrolled pretty significant disease in the sense that a high proportion of these patients had grade 3 or grade 4 acute graft versus host disease. We enrolled a high proportion of patients with liver and GI involvement as well. And so those were the major features which I took away when looking at patients that were enrolled into these cohorts. Next slide. So first of all, the primary objective was to evaluate the dosing, and what we had seen, and the way that we had actually approached this was by looking at receptor occupancy. With the dosing that we had administered, we had achieved a consistent receptor occupancy of 80% or greater with doses that were administered on this trial. Next slide. And so moving forward onto responses, we did see some promising responses with this age group. And so first of all, in the steroid refractory GVHD group, by day 28, we saw very modest responses at 33%. If we follow these patients longer, we did actually see pretty reasonable responses at 67%. What this may speak to is potentially underdosing actually within the steroid refractory group, with subsequent doses, perhaps exposure to drug increased, and so this led to sort of somewhat later improved responses in this cohort. Where we saw really pretty impressive results were in this treatment refractory acute graft versus host disease group. So we enrolled 12 patients, again, compared to historical control where our expectation was around 35%. 2/3 of patients by day 28 had a response when nihilizumab was added as their treatment refractory therapy. Next slide. In terms of adverse effects, we saw expected adverse effects for patients that were enrolled onto this trial. So with a focus on the multiple dose cohort patients, the most common or the most frequent adverse effects include effects on blood cell counts, so white count decrease, lymphocyte decrease, and we also saw infection as well in these patients, which we typically see in these agents, in patients who are on steroids and in the treatment refractory subgroup on potentially an additional agent as well. Next slide. So just to focus a little bit further, as we had mentioned, for steroid refractory acute graft versus host disease, ruxolitinib represents the most commonest treatment that is administered and was the most commonest treatment administered in patients enrolled in the treatment refractory cohort. And so we had patients who were treated with ruxolitinib. And I will point out that the median time to response that was reported in the REACH trials were about 5 days. And so, certainly these are patients who had received adequate exposure where we should start to see evidence of a response with ruxolitinib. What we had seen is in these patients, in over half of these patients, there was an improvement in staging and grading of graft versus host disease after nihilizumab administration. Next slide. I think in addition to seeing improvement in their GVH or stabilization in GVH, what was most impressive to me was we had mentioned that without stabilization or improvement of acute graft versus host disease, the median survival in this treatment refractory group in our survey was about 28 days. Because a large number of these patients improved with addition of nihilizumab, what we saw was in over half of these patients, there was, first of all, response, but there was survival at the 6-month time point. So I think this is actually one of the most impressive slides or the most impressive results that we generated from this study, which is to say in a highly morbid, highly fatal clinical situation of treatment refractory acute GVH, we were able to see stabilization and durability and survival in these patients. And this is due to both most likely response in acute graft versus host disease. Additionally, most likely the ability to taper corticosteroids, which leads to a lot of toxicities over time. And so this, I think, really speaks to potential activity of nihilizumab in this difficult to treat condition. Next slide. So in terms of our conclusions, so steroid refractory acute graft versus host disease continues to be an unmet need for patients who undergo allogeneic transplant. If you look over the REACH2 results, what you see is overall the responses with Jakafi or ruxolitinib are around 60%, but in severe grade 3 or grade 4, these responses are closer to 40%. Despite, additionally, With steroid refractory acute GVH, there are a fair number of patients who have persistent acute graft versus host disease requiring additional therapy. So in the REACH2 trial, that was about a quarter of patients. In clinical practice, probably closer to 30% or more. And our study represented the first study which systematically administered a novel agent into this population. We administered neolizumab after failing ruxolitinib in the majority of patients with activity, both responses in about 2/3 of patients and durability with high degree of survival at 6 months. So based on our study results, the future directions that we're considering primarily include treatment refractory acute graft versus host disease, could also be considered for high risk steroid refractory acute graft versus host disease as well. Next slide. And I think that's the end. All right, thank you so much. And I'll turn it back to Jesse.
Thank you, Dr. Abedin. We really appreciate you taking the time to go through that and really appreciate your leadership on this study. We hope that we can find a path to bring this to patients with treatment refractory disease. Next slide, please. So building on the successes of 168, we have been quite busy bringing 268 towards the clinic. And 268 is a more potent PSGO1 enhancer. And we've been working quite hard over the last year to bring 268 forward and excited to have the potential to dose patients early next year with 268. A quick comparison between 168 and 268. So 168 is a traditional IgG4 antibody with 2 binding epitopes to PSGO1. 268 is an Fc fusion protein. And it has 4 binding epitopes to the PSGO1 epitope that we're interested in binding for the desired effect. Based on preclinical data, 268 is significantly more potent than 168, somewhere in the range between 3 and 10 times more potent. The increased potency is going to allow us to administer the drug subcutaneously. And here is a quick comparison on the right-hand side, 168 and 268. We have improved bioavailability with 268. The half-life also appears to be longer based on the non-human primate studies that we've been conducting, as well as looking at the potency. We anticipate we'll be able to dose 268 somewhere between 1 and 2 milligrams per kilogram, which will allow us to administer 268 as a single subcutaneous injection. Currently, we're projecting the administration would be anywhere between every 2 weeks and monthly, which should make 268 much more patient-friendly than 168. In addition, in the non-human primate studies, 268 was shown to be well tolerated, dosing at up to 120 milligrams per kilogram weekly. And that was actually the NOAEL for 268. I mentioned we've been quite busy progressing 268 through the IND enabling studies. Happy to share that we were able to submit our IND approximately a month ago, and we received a rapid clearance and a may proceed from the FDA. This is going to allow us to initiate our first in-human study early next year. And based on that study, which I'll be going through the design in a subsequent slide, we will initiate our phase 2A study in ulcerative colitis. And then later next year, our plans are to initiate the phase 2B study in ulcerative colitis. Next slide, please. This is the planned clinical development program for 268. If we look on the left-hand side, the initial study will be in healthy volunteers. This will be a single ascending dose, multiple ascending dose approach. The initial dose for the single ascending dose will be 25 milligrams. We're utilizing fixed dosing, and that's what we'll be conducting in the clinic. We'll then rapidly progress up to 675 milligrams with the potential to add an additional cohort in the event that we want to identify a maximum tolerated dose. Based on the dosing of the 225 milligram dose, we will move into our multiple ascending dose platform, dosing 75 milligrams 4 times over a 28-day period, followed by a 225 milligram dose. The 225 milligram dose is effectively 3 milligrams per kilogram in a standard ulcerative colitis patient population. I mentioned that the 9 milligrams per kilogram was effective in the 168 studies. This is on our conservative estimate of a third more or 3 times greater potency, so a third of the dose. And we believe that 225 milligrams will likely be the high end of the dosing interval for our subsequent ulcerative colitis studies. Based on the successes of the first in human study, followed by our chronic toxicity study in non-human primates, we plan to initiate an ulcerative colitis study, effectively replicating the data that was provided for 168 in the ulcerative colitis patient population that was refractory to biologic therapies. The plan is to have a 12-week induction period, followed by a 40-week follow-up period. Based on the continued efforts in that 2A program, we will then subsequently initiate a 2B study. This will be a placebo-controlled study, approximately 250 participants. Currently, we're discussing a placebo comparison with 3 active doses, a low, medium, and high. The endpoints will be the standard endpoints with a 12-week induction period, followed by an open label extension, which will allow us to better assess the efficacy outcomes and the powering for our eventual phase 3 program. Next slide, please. And then building on the successes for 268 and ulcerative colitis, we believe that 268 has a broad opportunity across a wide range of autoimmune diseases. There's a number of diseases in chronic autoimmune disease that are driven by chronically activated T cells, and some of them are listed here. I am in the fortunate position as a drug developer of not only having preclinical evidence of this pathway's success, but also building on the clinical evidence. So we believe this pathway is de-risked across a broad range of autoimmune diseases, and we're very excited to bring this therapy into the clinic. With that, I will pass this back to Judy.
Thank you, Jesse, and thank you, Dr. Abedin. Next slide. So with the excitement about the novel mechanism, as well as the clinical validated data, what we are planning to do is to progress further for 268. As Jesse mentioned, we already got the IND acceptance and going into the phase 1 very soon. Entering the second part of 2024, we will have the phase 2A data, and towards the end of 2025, we should be able to complete the whole proof of concept phase 2B data. In order to develop a program, we are doing a Series B raise for $80 million to complete the whole entire study all the way till the end of phase 2B. And we are also working on talking to different partners to further expand into different indications that will help us to accelerate to bring this treatment to patients in many different indications. And for 168, with very promising data on the TR AGVHD, we are actively seeking for a partnership in order to progress to the planned pivotal trial next, and then also the commercialization. Next slide. So with that, I will wrap up the presentation, open up for the Q&A.
Thank you, Judy. At this time, we'll be conducting a question and answer session with our speakers. As a reminder, if you would like to submit a question, you may do so by using the Q&A text box at the bottom of the webcast player, or by emailing your questions to questions@lifesciadvisors.com. I'll now turn the call back over to you, Judy, to moderate the session. Thank you.
Thank you. There's a question coming, and I'll probably ask Dr. Abedin to first address, and then if Jesse has anything to add. The question is if it's possible, the response seen in the treatment refractory group was just a late response to Jakafi.
Yes, so, going back to our study, these are patients who on average initiated therapy with neolizumab about 2 weeks after ruxolitinib was initiated. And so certainly one of the things that this agent allowed for is continued exposure to ruxolitinib while we administered neolizumab. And so certainly some degree of the response could be attributed to continued exposure to ruxolitinib. That being said, with GVH that is not adequately controlled, continued exposure is a significant problem, with particularly grade 3 or grade 4 and lower GI GVHD, with the significant sort of morbidity associated with protein wasting enteropathy, malnutrition, increased risk for infection due to low antibody levels. These patients don't remain alive unless there's some stabilization. And so I think that certainly, continuing on ruxolitinib alone is typically not something that we are able to do, and weren't, when we use our sort of standard agents. And so I guess the short answer is yes, some of the effect could be due to continued exposure to ruxolitinib. However, we don't have a lot of success in continuing ruxolitinib in patients with persistent or worsening steroid refractory acute GVHD. And so feasibly, if that was just continued alone, there would be a high morbidity or mortality, without sort of turning to our next agent.
Thank you, Dr. Aberdeen. And we'll move on to the next question. This question is for Jesse. Do you think Entyvio and Humira are still the bar in UC and IBD more broadly? What does a competitive product profile look for you moving forward with more oral or novel biologics, either on the market or in late stage development?
Yeah, thank you, Judy, for the question. So whether or not Entyvio or Humira, for that matter, is going to be the standard of care or the bar, I think looking forward at the time that this product would be approved, the practical bar will likely be a biosimilar anti-TNF agent. So I think payers will require a step-through approach where a biosimilar anti-TNF will be required. And I think it's important for therapies that are now coming to market to be able to demonstrate effect in a biologic refractory patient population. And I think that's exactly what was seen with the initial 168 studies. In our future studies with 168, we do plan to enroll a substantial portion of our trials with this patient population, in that practically we do believe that payers will require a step-through approach. In addition, I think we have the opportunity, based on the work that we're doing in the early clinical programs and based on some initial looks at our 168 data, to identify the patient population through a biomarker that would be more responsive to our therapies. And then in addition, I do believe that the field is moving towards combination therapy. I think you're seeing this with some of the recent studies combining anti-TNF and IL-23 therapies. It's very encouraging to see the data with regards to the treatment refractory group from this acute GVHD patient population, where we successfully combined this mechanism with a JAK inhibitor and did so safely. So I think that's looking towards the future. I do believe that the standard of care, at least from a payer perspective, will be demonstrating effect in a biologic refractory patient population.
Thank you, Jesse. So the next question, I will take it. The question is, which T cell subset are impacted by your study drugs? Any impact seen to Tregs? So based on our data so far, we have seen the effects on the effector T cells. Based on our also immunophenotyping from the peripheral T cells, we have seen no impact to other type of T cells. In terms of Treg, there are reports, literature showing Treg, the PSGO1 mechanism can further enhance the Treg suppressing function. And we have some preliminary data supporting that as well, and we're in the process to further confirm that. The next question is, what are the theoretical risks of inhibiting activated T cells systematically with chronic treatment? Are there any other analog drug target that could support that, this will be safe for a long time. Maybe Jesse, you can take on this one.
Yeah, so looking at the mechanism of this PSGO1 agonism, this was really why I was excited about the potential of joining this company and working on this product. So going back to my initial drug development experience when I was working on Humira early in my career, we had multiple patients that would come into our clinical studies, and typically the onset of rheumatoid arthritis is in the mid-50s in a female patient population, oftentimes without treatment. After about 10 to 15 years, you would get effectively what was called a burnout of the disease in patients with rheumatoid arthritis. And the belief was, from a scientific perspective, where effectively you had exhausted this inflammatory pathway. I believe effectively what we're doing with PSGO1 agonism is we're pushing this inflammation to its natural resolution, effectively exhausting the immune system at an earlier state. And with that, I effectively believe that we're moving towards a more natural resolution of the inflammation. With regards to the long-term safety issues with this approach, what I'm able to confidently say is that 168 has been safe in patients that have been treated for with up to 9 milligrams per kilogram over a 12-week period, which gives us a lot of confidence moving 268 into the clinic. And with regards to long-term outcomes, I think that's just something that we'll have to see with the later phase studies.
Thank you, Jesse. The next question also related to that, and I will start addressing that. If anything, Jesse, you can add it, definitely feel free to chime in. So the question is, given PSGO1 is highly expressed on platelets, would you have any safety concern related to agonist antibody interaction with platelet? First of all, I think based on our own data, we haven't seen PSGO1 really highly expressed on platelets. Definitely there are some literature reports on that, but that's not the evidence we have seen that. But I think the ultimate question really is about the safety. And I echo what Jesse has been stated. The beauty about what we are working on now, we already have almost 200 patient data to show the safety. And I think even based on our recent study on the TR and SR AGVHD, we haven't seen any side effect, significant side effect to the patient. So we don't believe this mechanism with that target towards a late stage T cell, frankly speaking, really bringing much safer solution because it really tackle the problem where it should be addressed. Anything to add, Jesse?
Yeah, I agree. So looking at the clinical program with 168 with up to 200 patients dosed, we've not seen any issues with regards to interference with platelet activity. And from a science perspective, given the fact that our binding epitope is at the C-terminus of PSGO1, away from the primary activity of potential platelet interaction with, for example, endothelial cells, I don't anticipate anything from that perspective either.
Thank you, Jesse. So the next question, definitely I think we're open to maybe both Dr. Aberdeen and Jesse to answer that. So the question is, what kind of a placebo response would you have? Sorry. Let me just read that question again. Any early evidence that achieving a CR with 168 translate to improved durability or survival compared to patient achieve a PR?
I think certainly that depends on the organ system. So actually one of the analyses that we're currently performing are patients with lower GI-GVHD who achieve a PR. And so I would say in that situation, achieving a PR is probably not adequate in terms of significantly improving survival, because these patients continue to have malnutrition, continue to require TPN, continue to require steroids and so are at an increased risk of infection. So I would say in that context, achieving a PR is probably sub-sufficient. And then really achievement of a CR is what's necessary in order to see patients with a durable response as well as survival. Certainly other areas, and this is why PRs are considered to be more significant in chronic GVHD. If you have a PR, for instance, with upper GI GVH, if you have a PR in skin, these patients are less critically ill. If you have a PR in liver GVH as well, they're less critically ill than if you have versus sort of a PR in lower GI. So it's really context driven. But I think that generally speaking, a CR certainly indicates, that a patient will do better.
Thank you, Dr. Aberdeen. The next question, and I'll ask Jesse to take this one. What kind of placebo response would you have expected from the patient population in your phase 2A study? I assume that's for the UC.
Yeah, thank you for the question. So the placebo response that we would have expected in this patient population, given the fact that this was a biologic refractory patient population and probably the most severe biologic refractory patient population that I've seen in a UC study to date, we would have anticipated that the placebo response would have been somewhere between 0 and 3%. I know placebo responses have been creeping up recently over the last decade or so in ulcerative colitis studies. And I believe this is largely driven by the fact that in order to recruit patients, we as drug developers for inflammatory bowel disease have been forced to go to Central and Eastern Europe and study patients who are effectively naive to therapy. And I think that's why you've been seeing higher rates of placebo responses. Given the fact that our study was conducted in a multiple biologic refractory patient population in US only sites, I believe the placebo response would have been quite low. In addition, I think it was encouraging for us as IBD drug developers to see the Prometheus result with a very low placebo response. And I think this is largely driven by the fact that they had careful patient selectivity. And in addition, if you look at the countries where they recruited, some of those countries that have contributed to higher placebo responses were effectively excluded.
Yeah, the next question is a further clarification to understand our plan on the phase 2A and 2B. So maybe Jesse can elaborate. The question is, what sort of patient population would you target for your phase 2A and 2B studies with 268?
Yeah, so that's a good question. So effectively, what patient populations will be focusing on in the 2A as well as the 2B? In the 2A, our goal there is to effectively demonstrate effect in a 268 treated patient population similar to 168. So that'll be a group of patients that are refractory to biologics in a North American patient population. And we believe that that will be able to demonstrate the corollaries between 168 and 268 in a subcutaneous injection versus an IV infusion, so a more patient-friendly approach. For the 2B, in that larger placebo-controlled trial, likely what we'll do is a global study in patients who are both refractory to biologics as well as naive. Currently, we're discussing targeting approximately 25 to 30% biologic refractory patients in that study, and that will likely be replicated in the phase 3 program. But that will be a global study in both biologic refractory as well as naive patients.
Thank you, Jesse. So the next question is, after UC, what indication would you prioritize or think would be most promising? I think, Jesse, you love this question.
Yeah, and we've spent a lot of time discussing this, and I think coming into this company, into this program, it's really nice to be able to lean on the success of 168. So I'm quite excited about the potential of moving 268 into a rheumatologic patient population. And with regards to proof of concept, I think psoriatic arthritis, especially building on the 168 data, there may be a nice ability to run a study in that patient population, especially from the perspective of you have unique features in psoriatic arthritis. You have the synovial components, which if you're able to reduce activity in synovial inflammation, I think that's a nice read-through into rheumatoid arthritis. In addition, 30% of patients with psoriatic arthritis have associated psoriasis. So that's a nice read-through into skin conditions. And then finally, the enthesial component of psoriatic arthritis, being able to demonstrate an effect there has a nice read-through into patients with spondyloarthropathy. So I would like to move this therapy into rheumatologic indications.
Thank you, Jesse. The next question is, what other companies are currently working in this immune checkpoint enhancer space, and how do your program differentiate? So I can start. Definitely, we are very pleased to see ourselves as the leader in the immune checkpoint enhancer. With the success of the immune checkpoint inhibitor in the oncology field, and this definitely is the logical thinking for people thinking about turning this around. However, AltruBio started this way early, back to the early 2004, sorry. And so back then, we already starting building the data. The most advanced part for us really are be able to show the clinical data for up to 200 patients compared to other companies in this space like Eli Lilly, MiroBio, or Anapsys. We already have the clinical data to demonstrate the advancement. And towards the PSG01 in this novel way to target, like Jesse elaborate, from 168 to 268, how do we enhance that function by the molecular design? That's definitely also our advantage. So we're very pleased to see this kind of mechanism will be able to target exactly the problematic T cell and bring it down, should be ultimately what immunological disease that need. So we can take that lead in this space. The next question is, what is your sub-Q injection volume for 268? Is it a single injection? How long does it take to inject? Maybe Jesse, you can take this question.
Yeah, so thank you, Judy. So we're anticipating that we will be able to administer effective doses in our planned dose range between 1 to 2 mLs. So it'll be a standard single subcutaneous injection given to patients somewhere between every 2 weeks and monthly.
Thank you. Let's see, there's a next question coming. The next question is, this is probably for Dr. Aberdeen. So were there any of the SAEs seen attributable to the ALTB-168 neholizumab?
Yeah, so certainly lymphopenia, low white count, infection, with the addition of immune suppression, these would be SAEs that investigators felt likely were due to addition of this additional agent, in both the treatment refractory and the steroid refractory patients.
Thank you. We're open to the next question. The next question is, if narrowing to the treatment refractory population, is there a viable market? Would it be possible to target the steroid refractory population in combo? I can address a little bit about the market, and then maybe Dr. Aberdeen, you can make comments about targeting the steroid refractory. So in terms of the market, as Dr. Aberdeen pointed out, for the patient being treated by Roxo, we see 25 to 30% of the patients that really did not respond. So we consider that population is still somewhat significant. By looking at all the other available treatment, and this definitely 168 introduced additional, maybe actually one and only solution for TR for now. So we see that is still decent size of population we should address. Is that possible? We can move to the SR. Definitely, once we be able to demonstrate that, our belief is somehow it should be able to tempering also Roxo. But I'll reframe that comment. Maybe actually ask Dr. Aberdeen to comment on that.
I think in patients with steroid refractory acute GVH, these are patients that are typically in high dose steroids. They're initiated on ruxolitinib. And so, the immediate introduction of an agent, including natalizumab, which doesn't appear to have significant sort of infectious events associated with it, still increases risks. And so I think that this approach of initially observing a degree of response to ruxolitinib, particularly if it allows for some degree of mitigation of steroids, is just kind of a safer approach in patients. And so I think furthering, investigation and treatment refractory, if we particularly see that the safety signal is maintained, and we also see the signal in terms of efficacy, that would be sort of a sequence I would like to see before just moving this a lot up into the steroid refractory space.
That makes sense. Yeah. Thank you, Dr. Aberdeen. So the next question is, how do you think about target companies for partnering for 168 versus 268? I can probably start by saying, ideally, would love to have a partner would be as enthusiastic as us on both molecules. So that would be great. But if we will try to separate these two different target companies, we can do the partnership. For 168, definitely will be ideal to have the company already have the infrastructure in the hemolytic cancers space. So then that for the commercialization going into the marketing and sales, that will be much quicker. And for 268, definitely can apply broadly to all many immunological diseases. So that will be probably very applicable to all the big pharma and biotech. That's what I will say. That's maybe the subtle difference, but definitely we are very passionate about both molecules. So next question coming is, how does this mechanism compare to other agonists targeting PD-1, BTLA? So I can start, and if Jesse has anything to add, definitely feel free to put in your comment. So logically, as I mentioned, people were thinking about PD-1 works in the inhibition direction should work for the agonist direction. So that's a very logical thought, so as that's applicable to all other immune checkpoint regulators. I think the biggest difference really is we already have the clinical data and also for the phase 2 data. I think it's too early, at least, I mean, as a scientist, I will refer my comment about to say which mechanism or which key features of the enhancer should have. And we really need the more data to show, but we definitely have been studying the PSGO1 regulation, particularly using 168 in grand details. We know this mechanism, how that make PSGO1 works. I think that's a unique part about targeting PSGO1 and using 168 and 268. Yeah, so we think that's very promising, but the comparison yet need to be proven, seeing other targeting, how their data looks like. Anything to add, Jesse?
No, Judy, that was a very thorough response. I think I could add the benefit there.
Thank you. So the next question is, how does this, I'm sorry. Any other question? We don't see any question coming. So with that last question on our mechanism, I would like to thank you for joining us. With a special thanks to Dr. Aberdeen for the great presentation. And as a reminder, a replay of this call will be available on our website for 90 days. So with that, I will close the call and thank you and have a good day or good night wherever you are.